Photosynthesis. Photosynthesis is the process that occurs in plants, algae, and some kinds of bacteria that use the sun's light energy to chemically convert carbon, which is inorganic, into carbohydrates, which is organic, like sugars and starches. It will capture the energy of sunlight and the bonds of glucose. There are two main reactions in photosynthesis: light-dependent reactions and light-independent reactions. Light-dependent reactions occur in the green thylakoids within the chloroplast. The thylakoid also contains two photosystems. Light-dependent reactions begins when light splits water molecules in the process of photolysis. Light-independent reactions take place in the stroma of the chloroplast. These two processes will be further explained in detail later on.
Light-dependent reactions of photosynthesis. In this reaction, two high-energy compounds are created, ATP and NADPH. These two compounds have the effect of a large amount of reducing power. In this reaction, light energy is absorbed through the pigment of the thylakoid membrane. Pigment is a compound that absorbs certain wavelengths of light while reflecting others to give the plant its green color. Light energy is trapped in the pigment and then passed on to other molecules that produce ATP and NADPH. Photosystems are clusters within the thylakoid membrane that contain chlorophyll molecules, carotenoid molecules, and electron-accepting molecules. These components are responsible for absorbing and reflecting different wavelengths of energy. Next, the energy is passed on to the reaction center. The reaction center is where the electron has been raised to a higher energy level. From the reaction center, the electron moves to the electron acceptor that has been reduced because of the presence of the electron. Because of this, there will be a higher energy level. The electron is then replaced by a water molecule, so no more sunlight energy can be absorbed by photosystem number 2. The water molecule is then split in order from electrons, hydrogen atoms, and electron atoms so they can be released. Step 2. Next, the electron is transferred along an electron transport system. With each transfer, a small amount of energy is released in order to guide the hydrogen ions from the stroma to the inside of the thylakoid. For the energy that is released as an electron, it is passed through the electron transport system. It is then stored for a brief period of time in a hydrogen ion concentration gradient across the thylakoid membrane. There are more hydrogen ions in the thylakoid than there are in the stroma. In the hydrogen ion gradient, the energy is used to generate NADPH and ATP and to free phosphate groups. The third step occurs when step 1 and 2 happen. During this time, the light energy is absorbed by photosystem number 1. The energy is transferred to an area that is a reaction center where the electrons become excited and move around a lot. This excited electron will then pass to a high-energy electron acceptor. The lost energy from photosystem number 1 is now replaced by an electron that reached the end of the electron transport system back from photosystem number 2. Step 4. The last step involves the electron that was received by the electron acceptor from photosystem 1 in step 3. This electron is used to reduce NADP+ and form NADPH. The reducing power that is from NADPH will be used in the light-independent reactions. Chemosmosis. Chemosmosis is part of the process in making ATP. This is when the hydrogen ions are forced to move from the stroma to the thylakoid space. There is no going back from this process because the membrane is impermeable towards these charged particles. There is only one pathway for the hydrogen ions to move down their concentration gradient due to this. This pathway would be ATP synthase that is embedded in the thylakoid membrane. Since this pathway is linked to a mechanism, this bonds a free phosphate group to an ADP molecule to then form ATP. To do this, while the hydrogen ions mow down their concentration gradient through ATP synthase, that energy will create ATP molecules.
light-independent reactions of photosynthesis. This is when there is a good amount of NADPH and ATP that is required in the stroma of the chloroplast. The energy from there will be used to incorporate glucose in the presence or absence of light. The Calvin-Benson cycle is the series of reactions by which carbohydrates are synthesized. Here are the steps for the Calvin-Benson cycle. The first step in the Calvin-Benson cycle is fixing carbon dioxide. This involves carbon dioxide fixation, which is when the carbon atom in carbon dioxide is chemically bonded to a molecule in the stroma that already existed. The molecule contains a 5-carbon compound, which is ribulose biphosphate, RUBP for short. The result of this makes a 6-carbon compound, which means that it is unstable and will break immediately into 2, resulting in 2 3-carbon compounds. Step 2 is reduction. This is when the now new 3-carbon compounds are now in a low energy state. However, they need to be at a higher state. Therefore, to convert them to a higher state, they need to be activated by ATP and then reduced to NADPH. This result is called glyceraldehyde 3-phosphate, or PGAL for short. During their higher state, some of the PGAL molecules will leave the cycle and will be helpful towards making glucose. The remaining PGAL molecules are helpful for the next step of the process. The third and final step is replacing RUBP. This is when the remaining molecules of PGAL are used to make more RUBP. To break and reform chemical bonds to make the 5-carbon RUBP from PGAL requires ATP. To make one glucose molecule, you must complete the cycle 6 times. This means that one of the 12 PGAL molecules that are produced in 6 carbon cycles, 10 are used to regenerate RUBP and 2 make glucose.
Cellular respiration is the process that occurs in the mitochondria that is part of animals and plant cells, as well as other multicellular organisms, breaking down carbohydrates and any other energy-rich products that comes from things such as fats. What this does is generate molecules of ATP, which provides energy for cellular work. Cellular respiration releases the energy in glucose molecules by oxidizing glucose to carbon dioxide. This is when the electrons and hydrogen ions are removed from glucose. It releases energy and produces carbon dioxide and water.
Aerobic cellular respiration. This is the organisms that need oxygen. It involves the full respiratory pathway of glycolysis, the Krebs cycle, the ET chain, and chemiosmosis. For this process to occur, oxygen acts as the final hydrogen and electron acceptor at the end of the ET chain. Anaerobic cellular respiration. This is in the organisms where no oxygen is available. The process is more complex compared to the aerobic cellular respiration process due to the fact that this process is also known as fermentation. Fermentation will be further explained later on.
Glycolysis can occur in living cells. For some cells, this is the only source of energy, since it can proceed anaerobically, such as yeast. Glycolysis occurs in the cytoplasm of the cell, where it will begin its progressive breakdown of glucose. Therefore, it occurs outside the mitochondrion. Glycolysis means a process that splits glucose in 2. ATP is used at the start of this process. Once energy is added to glucose, it splits and 2 intermediate 3-carbon molecules are formed. ATP is incorporated and molecule of NAD+ is reduced to NADH. Overall, 4 molecules of ATP is produced, but only 2 molecules of ATP is used in the process. When glycolysis is complete, there are 2 identical 3-carbon molecules of pyruvate. For anaerobic cells, the process will finish in fermentation. Once there is the right amount of oxygen that is needed, pyruvate is transported into the matrix of mitochondria to prepare for the Krebs cycles. The preparation process where it goes from the glycolysis process to the Krebs cycle works like this. Pyruvate will lose a carbon atom in the form of CO2. It will then bond to a molecule called coenzyme A, also known as CoA. While this is happening, another NAD+ is reduced to NADH.
Krebs cycle. The purpose of the Krebs cycle is to progressively strip off more high-energy hydrogen pairs from glucose fuel fragments and send them to NAD+, and the electron transport chain will be used to make ATP. A 4-carbon compound picks up a group of 2-carbon compounds from acetyl-CoA and must be regenerated so it can pick up more 2-carbon groups. This will form 6-carbon citric acid. During this process, a 2-carbon group is added and 2 carbon atoms are fully oxidized to CO2. CO2 is a waste product by diffusion and exhalation. When the CO2 molecules are removed, it leaves behind 5 carbon and then 4 carbon fuel fragments. Most of the energy that is released when the carbon atoms are oxidized is turned into reduced power, but now in the form of NADH and another molecule that is called FADH2. During the Krebs cycle, there is 1 ATP molecule that is generated.
At this point, very little ATP has been produced up to now. Therefore, this is the stage where the vast majority of the ATP molecules in aerobic cellular respiration are produced. The components of the chain are protein pumps and carriers embedded in the inner mitochondrial membrane. High-energy NADH molecules have been produced during the process of glycolysis transition in the Krebs cycle, when NAD+ is reduced by hydrogen atoms. During the final step of aerobic cellular respiration, NADH releases an H+ ion into the mitochondrial matrix and 2 high-energy electrons enters the ETC. Energy is released in the small controlled amount as electrons are passed from one electron to another. This energy is used to pump hydrogen ions across the membrane from the matrix to the intermembrane space. The ions in the intermembrane space that have built up then creates a hydrogen ion and concentration gradient. These ions can diffuse back across the membrane but only through channels created by the enzyme ATP synthase. This enzyme will use the energy of the concentration gradient to link together a phosphate group to ADP, which will form ATP. This process is also known as chemiomosis. Oxygen is the final electron-accepting molecule in the ETC. The oxygen accepts electrons and hydrogen ions, forming H2O. ETC produces 36 ATP molecule.
Fermentation. Fermentation is the chemical breakdown of carbohydrates, starches, and sugars by bacteria or other microorganisms. This exothermic reaction, releasing of heat, involves bubbling and fizzing. An example of fermentation is the creation of alcoholic beverages such as wine. It is the process that follows glycolysis for cells where oxygen is not present. It is a metabolic pathway that includes glycolysis and 1 or 2 reactions in which NADH is oxidized to NAD+ by reducing pyruvate to other compounds. Fermentation occurs in the cytoplasm of the cell. There are 2 types of fermentation: lactate fermentation and ethanol fermentation.
There are some single-celled organisms as well as some animal cells that are temporarily without oxygen that carries out lactate fermentation. NADH is used to convert pyruvate to a molecule of lactate. The resulting of NAD+ is recycled in order to continue this process. An example for where this process can occur is your muscle cells when they are working simultaneously. The demand for energy exceeds what can be produced aerobically. Glycolysis is then increased to a point where it exceeds the oxygen supply. 2 molecules of pyruvic acid uses NADH to form lactic acid.
Ethanol fermentation. For some organisms, they are able to function aerobically as well as anaerobically. Ethanol fermentation is carried out when they function anaerobically. An example of ethanol fermentation would be with yeast.
What are 2 differences between your model and the real structures? One difference between our model and the real structure is the size. The actual cell would be much smaller than our model due to the fact that the chloroplast alone is 4 UM by 6 UM in diameter, and the mitochondria is 0.5 UM by 1 UM in diameter and 4 to 5 UM in length. Other components of the cell are also similar in size. Altogether, the size cell of the whole is microscopic. Our model is as big as a shoebox, so there is a significant difference. Another difference is the materials that we used to make the cell. We used pipe cleaners, styrofoam balls, water bottles, and Play-Doh to make all the components. An actual cell is made of molecules that include proteins, lipids, and carbohydrates.
What are 2 design challenges that had to be overcome while planning your model? The first design challenge was figuring out what materials we wanted to use to make the model and getting all the materials together. Our second design challenge was actually building the parts of the model and trying to make the cell look as realistic as possible.
If you started your project again, what would you do differently? If we did this project again, we would try to be more original and make it with food instead of Play-Doh, water bottles, pipe cleaners, and a shoebox. Another thing we would do differently is start the model earlier so we would have more time to work on other parts of the project.